A very lovely good evening to all of you. I am Dr. Pooja Johari, your biochemistry educator. So since today it's the first live session with you guys, so I will be starting up with the chemistry of carbohydrate. Right? Now before starting up with carbohydrates, as it is clear from the word itself, carbohydrates are what? They are the hydrates of carbon, isn't it? Hydrates of carbon. So there are so many definitions about these carbs. The earliest system of classifying carbohydrates was that carbohydrates are the ones which have carbon, hydrogen and oxygen in the ratio 1:2:1. But nowadays, the widely accepted classification of carbs is that they are polyhydroxy derivatives of aldehydes and ketones. Right? They are polyhydroxy derivatives of aldehydes and ketones, and on hydrolysis, they give aldehydes and ketones too. Since this is a live session, I am just waiting if you guys have any problem. So you people don't hesitate to ask me in the chat box. Carbohydrates is a simple topic, but it is a complex one also. So we are here to tell you the complexity of carbohydrates plus the simplicity of carbohydrates also. Right? So these are what? Polyhydroxy derivatives of aldehydes and ketones, which on hydrolysis will give these aldehydes and ketones too. So that is why the first way of classifying them is that they are of two classes. If they have aldehyde as the functional group, they are known as aldoses. And if they have ketone as the functional group, they are known as ketoses. And the interesting part about these is that these aldoses and ketoses are linked as isomers with respect to one another. And this is a very interesting factor which takes place in the body. I can see a Tamil speaker is, beta, this is carbohydrates is a very generalized topic. It is for first year MBBS students. It is for those students also who are preparing for their NEET exam also, those who are preparing for their INI CET. So it is a very common topic for all the medical students. In fact, this is an important topic for those, future aspirant doctors also who have taken medical as their subject for class in class 11th and 12th. Right? So aldoses and ketosis. So aldoses, yeah, good evening. Nagesh, how are you? So can anybody tell me aldoses, what will be the principal functional group in this? The principal functional group in aldoses will be always a aldehyde. Right? And for ketosis, it will be a ketone. And it is so interesting that aldoses and ketosis are linked as isomers, and this reaction takes place in the liver also. Now, this is one way of classifying carbs. Now see, another way of classifying these carbs is depending upon the number of carbon atoms. So we always start a carbohydrate having 3 carbon atoms, which is known as a triose sugar. And the example of a aldotriose is glyceraldehyde. Right? And an example of aldotriose is glyceraldehyde, and ketotriose is dihydroxyacid. Right, very good Tamil speaker. I really liked your name, Tamil speaker. Okay, but you are speaking in English. That's nice. Right? Then coming over to if it has 4 carbon atoms, that means it will be a tetrose. An example of a aldotetrose is erythrose. and ketotetrose is erythritulose. And again, the interesting part is erythrose and erythritulose. They are linked as isomers. And these are the most important intermediates of energy. Because why we eat food? Obviously, carbohydrates are the universal sources of energy. You know, we people are fond of eating carbohydrates in the form of bread is there, then rotis are there, then these burgers and all. They are all carbs, right? So these are what? These are energy giving. Then coming over to 5 carbon atoms, it is pentosis. Now again, example of pentosis, if I talk about aldopentose, the example is ribose. If I talk about ketopentose, the example is ribulose. Now all of you are familiar with DNA and RNA. Ribose is the major constituent of DNA and RNA. In DNA it is deoxyribose, in RNA it is ribose sugar. Again, ribose and ribulose are connected as isomers with respect to one another. Then coming over to the next one, 6 carbon atom hexosis. Aldohexose is glucose, right? And isomer of glucose is fructose. And fructose has more sweetening index than glucose. Fructose is a more sweeter sugar than glucose. Again, glucose and fructose are linked as isomers. So this is one way of classifying carbohydrates as per their, first of all, the functional groups. You can see aldose and ketose, and then as per the number of carbon atoms. Then another way of classifying these carbohydrates is as per their units, which means that as per their units, they are divisible into 4 major classes. Right? The most important, the simplest one is monosaccharide. Mono means single. Saccharide, actually saccharide is derived from, because sugar is derived from sugar cane. And the botanical name of sugar cane is saccharum. So that is why we always write saccharides. Mono means single. Then if we talk about 2, it is disaccharide. Is any sugar have more than one aldehyde and ketonic group? Yes, of course. But the principal group will remain the same. Principal group will be an aldose and ketose only. It cannot change. Depending upon the carbon atoms will change. Tamil speaker, can I get your original name please? If you don't mind. Or otherwise, can I call you TS? Okay, Sanjay. So, Sanjay, beta, what I'm trying to tell you here is, your question is, aldehyde is the main functional group. I'll just show you the structures also, open chain structure. Then you will see aldehyde, CHO cannot be 2. But yes, CHO and CH2OH can be there. So, this is an aldehyde and this is what? This is a alcoholic group. Clear? So, yeah, so depending upon the number of units, monosaccharides are there, then disaccharides are there. Again, it is clear, dimane 2. Then oligosaccharides. Then comes polysaccharide. So in this section of chemistry of carbohydrates, we are going to discuss each and every unit one by one. What is this biomedical significance? How much we have to eat, in what quantity we have to eat. Because ultimately, we are studying biochemistry. Biochemistry, it is the chemistry of the biomolecules. What is happening inside our body when we are having this much of carbohydrate? Because nowadays, this diet plays a very, very important role in our body. In fact, diet is the one which is actually regulating the entire body, isn't it? So how much carbs we have to eat, how much glucose we have to eat, how much fructose we can, because we cannot go to the shop and we can ask for glucose. No, we can ask only for a particular dish. How many carbs it is containing, how much calories we are consuming, what role it has to play in the body. That is all what we are going to cover in this. So chemistry is very, very important to understand biochemistry. Because I have seen most of the students saying biochemistry is a volatile subject, it just evaporates, it just goes off. If you try to understand the chemistry, because whatever is happening, it is happening inside the body. So automatically, it's all your perception of mind, how you take it. So it is the chemistry, what is happening inside the body, because if anything goes wrong, it leads to diseases. And being a doctor, we should know what kind of disease it leads to so that we can regulate. Right? So that is what we are going to study. What are monosaccharides? What are disaccharides? What are oligosaccharides? What are polysaccharides? So mono mane single, as it is clear from the word itself. Di mane when two monosaccharides combine. Oligo mane when 3 to 7 monosaccharides combine, and poly mane many, when more than 7 combine. So these are the two different ways of classifying the carbohydrate. Now the topic of today's session is chemistry. So we will be studying the chemistry of, first of all, the monosaccharide. Right? Now again, what all we are going to study in the chemistry? First of all, what are the examples? The examples of monosaccharides are glucose, fructose, mannose, galactose. Right? What is the chemistry means what are their structures, what kind of isomerism is present in them. Because always remember, here we are talking about the body, and always remember one thing, everything tends to go in a stable conformation. Like if right now you are attending my lecture, some people must be just attending the lecture like sitting. Some means everybody tries to go in a comfortable position. Some people like listening to the lecture by lying down. Some people like listening to the lectures in a proper way, like studying on a table and a chair. So same is the case with these all glucose, fructose, mannose and galactose. They always tend to go in a stable conformation. That is what is the structure and isomerism all about. Then we are going to study the properties. Yes, I will come to the optical rotation behavior. Optical rotation will come in the isomerism. Yes, Apple Mania, what is your actual name? Can you please tell me your actual name? If you don't mind. Because Apple Mania seems to be, because I am also using an Apple thing, so it will create confusion in my mind. So please share your actual name. Yeah, it's my pleasure, Sanjay. Okay, so optical rotation we'll be discussing in isomerism. It will come, right? So structures we are going to study, isomerism, because everything depends on the structure. And then what are the properties? What are the chemical properties? Mainly we will be focusing on the chemical properties, right? Sagar. Okay, Sagar, right? So now first of all, we will start up with the isomerism only. By isomers, all of you are familiar with isomers. You've been studying isomers from donkey's years. Isomers are what? The isomers are defined as those compounds which have a same molecular formula but different structural form, isn't it? Let's say, for example, if I talk about glucose and fructose, C6H12O6. In fact, if I talk about all, they all have C6H12O6. But what is the difference? Because whenever we talk about the body, there will be a difference. There will be a difference in the metabolism of glucose, in fructose, in mannose and galactose. Right? So isomerism is simple. This is a widely accepted definition. Just keep it that way. Same molecular formula but different structural. formula. If they have a different structural formula, they are known as structural isomers. It is simple, very plain and simple. Structural isomers, example is, you can say glucose, fructose, because glucose is what? It is having an aldehyde as the functional group. So I can say glucose is what? Glucose is a aldohexose. And fructose is a aldohexose. Right? Yes, they are the functional. Very good, Sanjay. They are the functional isomers also. They are the structural isomers also. Why functional? Because there is a change in the functional. Right? Then second important type of isomerism, which is actually very, very important from examination point of view, also stereoisomerism. Now see, stereo. Stereo means isomerism, simple, same definition. But if they have a different orientation as per in the space, that comes from stereoisomerism. Because if I show you the structure of glucose, see, glucose is having this structure. As such, you don't have to mug up the structures. In case if you can mug up, well and good. But I won't suggest you ki, don't get panic if you don't remember the structures. Because ultimately, once you get anything in the body, what is happening inside the body, that you have to remember. Okay. So this is 1, 2, 3, 1, 2, 3, 4, 5, 6. Right? This is having an aldehyde, as it is clear. 2, 3, 4, 5 and 6. Now see, just for your this thing, hexose sugar, 6 carbon atoms, right? Aldohexose, aldehyde as a functional group. So it is a aldohexose. Okay? So this is the structure of glucose. Now you must have seen, all of you have studied, chemistry, you've been studying chemistry from class 9. So you must have studied this D-glucose. We write it like this. So from where this D comes from? Have you ever given a thought? From what do you mean by this D? I'll ask you, Sanjay, Sagar, can anybody tell me? Now I know you will say dextro or levorotatory. This D is actually the orientation in the space, and these are known as stereoisomers. I'll come to that. Now, how can we, just by looking at the structure of glucose, we cannot say D-glucose, isn't it? We need to have a standard molecule with which we can compare glucose and assign a configuration. So that standard molecule is glycerol aldehyde. Just try to understand this concept. It is very simple. Glycerol aldehyde, now tell me. Yes, very good, Sanjay. Fifth carbon atom has OH on the right hand side. But how we came to know that it is D? We have to compare this structure with something. Cis and trans is altogether different. Don't go haphazard, just move step by step. This is same side, trans is opposite side. I'm coming to that. Here we are talking only about stereoisomerism. Okay, so don't get deflected by different terminology. I'll just come to that. See, if we talk about glycerol aldehyde, all of you know, yes, very good, Nagesh. Right, so glycerol aldehyde, if I talk about, I can write the structure of glycerol aldehyde like this. Yes, very good, and CH2OH. If I say, first of all, let's look at this carbon atom. This carbon atom. Okay, you know what is the significance of this carbon atom? This is a chiral carbon atom, first of all. Now, what is a chiral carbon atom? It is also known as an asymmetric carbon atom. Now see, with this D terminology only, there will be so many terms which will come up. Right? So this is a chiral carbon atom. This is a asymmetric carbon atom. It is defined as that carbon atom on which all the 4 substituents attached are different. 4 substituents attached are different. So if they are different, what is the significance of that? Right? They are going to form a mirror image which will be non-superimposed. Isn't it? If I make a mirror image of this, I can write it like this. Yes, very good. Mirror image will be there, but it will be non-superimposable. And always remember one thing, that those isomers which can form a non-superimposable mirror image, non-superimposable mirror image, they are known as what? They are known as, yes, very good Nagesh, enantiomers. So always remember, whenever enantiomers we are talking about, first thing we have, what will come into our mind? They will have a chiral carbon atom. Chiral carbon atom means all the 4 substituents attached will be different, or they are going to form a non-superimposable mirror image. But my question was, this was okay about chirality. My question was, why we call glucose as D-glucose? Now see, what happens is, in chemistry there is a term called Kiliani-Fisher synthesis, which means that from a 3 carbon atom sugar we can generate up to 6 or 7. So here what we are doing is, the second carbon atom, I'll just repeat it again. This second carbon atom is going to behave as a second last carbon atom in successive sugars. Means if glucose has 6 carbon atoms, the 5th carbon atom, which is the second last, we will stick to the position where OH group is present there. Now here you can see that OH group is present on the right hand side. Here OH group is present on the right hand side. So this form of glyceraldehyde is known as D-glyceral. And when OH group is on the left hand side, it is known as L-glyceraldehyde. Right? So one thing is, let's just revise it. When OH group is on the right hand side in glyceraldehyde, we call it as capital D glyceraldehyde. When OH group is on the left hand side in glyceraldehyde, we call it as L-glyceraldehyde. As we move to successive sugars, like 3 carbon atoms we are discussing, 4, 5, 6, 7 and so on, the second last carbon atom will always be given preference. So if we compare the structure of glyceraldehyde with this, we see that OH group is on the right hand side, and that is why we call glucose as D. And this is again a very interesting thing. In hospitals also we get glucose in the form of dextrose. So always remember, D for dextrose, D for D-glucose. A question arises, why D-glucose? Because glucose is stable in. So we always say that it is always D. I am not saying L-glucose does not exist. L-glucose do exist, but glucose is most stable in D form. So whenever we talk about a body, everything tends to go in a stable conformation. So obviously glucose will be stable in D form. That is why it is known as D. And these are what? These are stereoisomers. So we can say that stereoisomers made D and L are enantiomers, right? Or they are stereoisomers. Now the question is a very interesting question. Let me see. What's the origin of enantiomer specific chiral Interactions in molecules. Beta see, chiral carbon atom is the one, just now I told you, as you have studied, you must have studied in chemistry about rectus and sinistrus configuration, E and Z. There are so many configurations, like whether it is right, whether it is left. So what is this all about? It is all about the stability. What is the stability of the molecule? Means in what state it can exist for a longer time. That is what is chirality all about. That is what is D and L isomers all about. Is it clear, Sanjay? Okay, so always remember one, yeah, because you know, if I talk about chemistry, you know, you have studied about chair conformation is there, board conformation is there, cis is there, trans is there. So everything tends to fit in a stable conformation. So whenever we talk about glucose, it has to be in capital D form. Similarly, if we talk about fructose is stable in. Okay, one more hint I will give you. Okay, my pleasure, Apple Mania AM. Okay, so what we can say is fructose is levorotatory. It is stable in N form. We can put it that way also. But don't get confused between small d, small l and capital D and capital L. This I just want to highlight again and again. Whenever we talk about capital D and capital L, they are in response to the orientation in the space. Always remember. So we always talk about taking glyceryl aldehyde as a reference. So it is always capital D and capital L. This same thing you are going to. My pleasure, such a big compliment you have given me. Thank you so much. Right? So Apple Mania has given me mother of biochemistry. Thank you so much. So sweet of you. You made my day by saying this compliment. Okay. So always remember stereoisomers in space. Now I'm coming to another term, optical isomer. Now see, in science, if we see na, 99% of the problem is solved by just looking at the spelling only. By looking at the word, what the word has to say. Optical. Optical something related to. Isn't it? Stereo something related to space. So optical isomers definition would be the same. But optical means in terms of optical rotation. Right? Now optical rotation, how we measure? There is an instrument which measures. It is a polarimeter. This is again a very important question. Most of the students get confused between different type of meters, especially MBBS students. Calorimeter is there, calorimeter is there, polarimeter is there. So see, again you have to go to the spellings. Calorimeter means measuring the color. Because I have seen if the pronunciation is wrong, the entire thing goes wrong. Calorimeter is measurement of the calories. So there is a difference between a color and a calorie, isn't it? So polarimeter means optical rotation. So if a molecule is rotating the plane of polarized light in a right direction, if it is rotating, I'll repeat it again. If a molecule is rotating the plane of polarized light in the right direction, we call it as dextrorotatory, denoted by small d or plus sign. Right? And if it is rotating in the left direction, again depending upon the stability of the molecules, orientation of the molecules, it is levorotatory or denoted by small l and negative. Right? So these are what? So I can say D and L are what? They are optical isomers. Right? And if I talk about D and L, means capital D and L, they are stereoisomers. This is the basic. Now another twist is there if we mix the two. If I say mix means Now the term dextrose, yes, very good question. Dextrose is going to represent capital D because glucose is most stable. I am telling you again and again, D means dextrose, D means capital D or D glucose. Just remember it like, right? Now, if we mix the two, means what are we doing? We are making a mixture, that means D and L. Okay, so if we talk about the optical rotation, the optical rotation means positive will nullify the negative. So this will be optically inactive, one thing, right? And when it is a mixture, it is known as a racemic mixture. So always remember, racemic mixture is optically inactive. This is also a very, very important question. Racemic mixture is optically inactive. But if we compare small d and small l, they are optically, because they are optical isomers, so they will be optically active. Right? Now the difference between optical isomers and stereoisomers is clear to all of you. This is very, very important. Right? Optical isomers वो होते हैं in which you can say they have a tendency to rotate the plane of polarization, whether it is in right direction, whether it is direction. And stereoisomers they are always measured in terms of their position of the OH group. And I told you in successive carbohydrates, suppose if I talk about a 7 carbon atom sugar, which is pseudoheptulose, you have to see the second last carbon atom, wherever OH is, whether it is on the right hand side, whether it is on the left hand side. So suppose if it comes as an image based questions, वैसे chances are very bleak. So you have to always focus on the second last carbon atom. Okay, so this is a very important property of monosaccharides, that they have a tendency of undergoing isomerism, whether it is stereoisomerism, whether it is optical isomerism. Because here we are mainly thank you so much. I am also sending love from India. Thank you so much. Dr. Allah, we have just finished up with the classification and I have just discussed about the isomerism. So if you have any problem, you can ask me now. Abhi, we have just started up with a very simple topic. Okay. So these are what optical isomers and stereoisomers. After this, because here mainly we will be concentrating on the biochemical aspects. Otherwise, if I start teaching isomerism, it will take whole night, it won't finish also. But here we have to only stick to that isomers which play a very, very important role in our body. Right? Then moving on, after this, there are two very, very important terms. One is anomers and second is epimers. Right? Up now, what are anomers? Anomers are defined as, yes, very good. Anomers, very good, Nagesh. Anomers are defined as those isomers. Now see how we differentiate. Isomers which differ only at C1, which is bearing the functional group. So how do we define anomers? Anomers are defined as those isomers which will be differing only at carbon 1. Right? And carbon 1, by chance, it represents the functional group. Okay? So now what happens is, and if we take the definition of epimers, yes, Dr. Allah, this is whatever I am teaching you in biochemistry, this will be more than enough for a first year MBBS student. This I can give you in guarantee. Because I am focusing on each and every minor topic. Obviously, major topics though I will be touching up, but minor topics also I am discussing in the classes. So you can tell your other friends also to attend the classes. These classes will be very, very beneficial for all of you. Okay. So anomers differing at C1. Epimers are the isomers which differ at any other position except C1. To see how interesting it is. If they are differing at C1 position, they will be known as anomers. Now, if they are differing at any other position, be it C2, be it C3, C4, C5, they will be known as epimers. Okay, so this is the basic difference. Now, I'll just give you a, little bit knowledge about the structures, because again, I am telling you, don't focus on the structures more, but little bit knowledge you should have, because starting say this is your just beginning of the career. You people have already studied chemistry in your class 11th and 12th. So if you make a chemistry base for biochemistry, you won't find any problem in understanding the subject. And obviously you will be focused to what is happening inside the body. So it is more interesting. Okay. So first of all, if I talk about, now see, all of you know about this. I'll just write the structure of glucose again. 1, 2, 3, 4, 5 and 6. ठीक है, OH. Third position पे always remember OH is here. H, H, OH, H, OH. ठीक है. Now see, you must have studied about acetals, ketals, isn't it? What is the basic difference between an aldehyde and a ketone? So many things you have studied in your, maybe in 11th or 12th. Now, if this aldehyde is there, I can write this aldehyde like this, CH double bond O. Similarly, I can write it like this also. Rest I won't repeat, it is but obvious. Right? This is known as an open chain structure of glue. Now, just now I told you everything tries to go in a stable conformation. So what is happening is there is a reaction between the first carbon atom and the fifth carbon atom. So reaction means loss of a molecule of water, which we know, which we call as condensation also. So I can say that reaction can be between first and fifth from this side, and reaction can be this side. Isn't it? Wherever double bond O is there, because water is what? H2O. So if the reaction is like this, or if I say loss of water molecule is from here, I can say that it will have this type of structure. This will be the first carbon atom. If I say it is like this, it will be like this. Right? Rest everything will remain the same. So this will behave as the first carbon atom, this will behave as the second carbon atom, third, fourth, fifth and HH. See Sanjay, beta, there is no confusion. Just remember one thing, hemiacetals and acetals. Hemi means partial acetals, acetals means full acetal. So that depends on the functional group, how the reaction is taking place. Because if you remember, if I talk about the ketonic group is always placed like this. If you remember, because ketone is never given first position. Carbonyl carbon is having second group. So depending upon that, acetals and ketones are produced. But that is not a part of biochemistry, so we won't be touching that aspect. Otherwise, you people will get confused. Okay, just remember one thing. There is lactam ring formation. Okay, so it will be like either from this side or this side. So change will be only at first position. Rest everything will be same. Matlab rest chalo, I'll just write it. Once I'm just writing it. Chalo, I'll write it here also. But even if try to make these structures at home, it's very easy. No complexity is there. What I'm trying to tell you here is this is a resonance, basically resonating structure. Double bond O is present here, double bond O is present here. If the reaction is like this will result. If the reaction is like this will result. Or in a simplest language, the change will be only at carbon 1. You can see, if I talk about this carbon 1, you can see that difference kahan pe hai? Difference is only at the OH position, isn't it? Now see, if OH group is below or if OH group is placed downwards, we call it as alpha D. Very, very important. And if OH group is placed upwards, we call it as beta glucose. So now you tell me the change is at which carbon atom? If I talk about alpha and beta, by just looking at the structure, can you identify the change is taking place at which carbon atom? 1, 2, 3, 4, 5, 6. Yes, very good, Nagesh. It's at 1, isn't it? Sanjay, Sagar, फर्स्ट पोजीशन पे चेंज है, फर्स्ट कार्बन एटम पे चेंज है. फर्स्ट कार्बन एटम पे इफ यू लुक एट द राइट हैंड साइड, OH group is placed downwards. I call it as alpha D glucose. If OH group is placed upwards, it is beta D glucose. Now coming over to this I will say it is a open chain form of glucose. So can I say that open chain can be converted into alpha glucose, open chain can be converted into beta glucose, or all the three forms are interconvertible with respect to one another? Yes or no? Bolo? See, this you have to, this is a very simple concept. बहुत सिंपल है. देखो इसमें कुछ नहीं है. द रिएक्शन इज टेकिंग प्लेस ओनली एट द फर्स्ट कार्बन एटम. दैट इज व्हाट आई टोल्ड यू. नो एनोमर्स, एनोमर्स क्या होते हैं? एनोमर्स आर द आइसोमर्स विच आर डिफरिंग ओनली एट C1. एंड C1 क्या है? C1 इज रिप्रेजेंटिंग द फंक्शनल ग्रुप. अब इस फंक्शनल ग्रुप में इफ वाटर मॉलिक्यूल इज लॉस्ट फ्रॉम द राइट हैंड साइड और इफ द वाटर मॉलिक्यूल इज लॉस्ट फ्रॉम द लेफ्ट हैंड साइड, इट विल रिजल्ट इनटू ईदर अल्फा डी ग्लूकोज और बीटा डी ग्लूकोज. So my point here is that alpha D glucose and beta D glucose are differing only at C1, so they are known as anom. Now you tell me if anomers are there, whether this C1 is free or not. Free means whether it is free to react with something or not. Yes or no? I want an answer from you people. Don't hesitate. Whatever is there in your mind, just speak up. I just want to see how much you are understanding. My question is, I'll just repeat the question. My question is, just now I told you, anomers are the isomers which are differing only at C1 position. C1 is the functional group. Now my question is, whether this C1 is free or not. Whether the functional group is free or not. Arey, you can see the structures also. Looking at the structure, can you tell me whether it is free? Very good. Nagesh has said yes. Sagar, Sanjay, Dr. Allah, say something better. Whatever is there in your mind, just speak up. That is what is the advantage of a live session. You can clarify your doubts there and then. Chalo, I'll tell you, they have a free functional. So let's put it that way. Anomers are those isomers which differ only at C1 position, number 1, and they have a free functional group or a free anomeric carbon atom. And those sugars which have a free anomeric carbon atom, they are known as a reducing. Very, very important. अब देखो from isomerism, we started with isomerism only and we have come to reducing sugars. Now I'll tell you what is the significance of these reducing sugars. You know reducing sugars की क्या significance है हमारी body में? These are one of the very important abnormal constituents of urine. If reducing sugars appear in urine, it is a clear indication of glycosuria, which can lead to diabetes mellitus, which is very prevalent nowadays. So you can see how chemistry mixes up with biochemistry. So isn't it interesting? Right. So anomers kya hai? Anomers are simply the isomers which are differing only at, just remember this, okay? And then they have a free functional group, and those isomers which have a free functional group, they are first of all known as anomers, and secondly they are known as reducing sugar. So alpha and beta are linked as anomers. Then coming over to epimers. Now again there is a confusion. I'll just try to sort out that confusion. Epimers की definition I told you that they will differ at any other position except C1. C1 पे differ करेंगे तो वो anomers होंगे. Okay. If they are differing at C2, they will be epimers. If they are differing at C3, again they will be epimers. C4, C5. Now what are the examples of these? If I talk about C2, yes, very good. Galactose is, yeah, I am coming to that. Yes. C2 may always remember mannose is a C2 epimer of glue. Can anybody tell me C3? Allose. Very good. Nagesh, allose, again C3 epimer of glucose. C4, galactose. C5, let's see. C5, can anybody tell me C5? Okay, I'll tell you idose. Idose is a C5 epimer of glucose. And you know, all these reactions, okay, all these reactions takes place in the liver. This galactose is converted into glucose in the liver. And if the enzyme which is, see, if in our body the reaction is taking place, जैसे chemistry में हम क्या बोलते हैं, catalyst is the one which is increasing the rate of a reaction. In our body, the work is taken over by the enzyme. And if that particular enzyme is absent in the body, it will lead to diseases. And same thing happens if galactose is not converted to glucose. What will happen? It will lead to a very serious disorder, which is galactosemia, which we will be again studying in detail in the metabolic. Right? So these are what you can see. Simple. C2, C3, C4, C5, not on C1. Up now tell me, these epimers, can they be reducing sugars? Yes or no? Let me see how much chemistry you people. My question is, anomers to you know, C1 pe differ kar rahe. That also you know, anomers which have a free functional, yeah, you can say which have C1 free, they are known as reducing sugars. My question is, can epimers behave as reducing sugars or not? Why no? They won't have a C1 free. Chalo, I'll tell you, epimers also will be reducing sugar. Now, why they will be reducing sugars? Because reducing sugars की definition क्या है? They have C1 free. Okay, you got my point. So, see, okay, see, that is depends. That is not ring form. Actually, that is a cyclic form. This is a open chain form. I'll just show you. This is a open chain form structure of glucose. This is a cyclic. इसको हम बोलते हैं, this is known as Fisher projection. This is known as Haworth projection. Okay, and depending, coming over to this, देखो, all have a individual identity. Anomers are differing at C1. Okay, alpha and beta. Reducing sugars are the ones which have free functional group, and functional group is always positioned at C1. Now coming over to epimers, epimers kya hai? These are the isomers which are differing at any other position except C1. So C1 will be the same. Whether it is alpha and beta, that is something else. Okay, but they will have C1 free, so they will be reducing sugars, isn't it? Now you got my point. Nagesh, is it clear now? So I can say that epimers will also be reducing sugars provided C1 is free. If C1 free nahi hai, to they won't be reducing sugars. And always remember, all sugars are reducing. All sugars are reducing except sucrose. But just remember this bottom line. One more is there, trehalose, but here we will be mainly focusing on sucrose because all of you are familiar with sucrose, which is the common table sugar. So irrespective of anomers, epimers, all are reducing. Even starch is a reducing sugar, right? Even though it has one reducing end and the other non-reducing end. So these are what is idose. You can say that beta, yes, cyclic L-glucose, the only difference will be in the fifth position. Because if you see the fifth position, here CH2OH is there. It will be changed there. Right? So, here what we got to know is anomers, epimers and reducing sugar. Now, another important concept, since we are dealing with the properties, a very interesting property of the reducing sugars is that they have a tendency to undergo mutarotation. Muta means rotation means, you know, rotate karna. Muta means within itself. So if a molecule, or you can say कि whenever we talk about the solution, right, and this solution is allowed to rotate within itself, it will change from the open chain form to alpha form, or it can change to beta. So we can say that all the reducing sugars have a tendency to undergo mutarotation. All reducing sugars have a tendency to undergo mutarotation. So I can say that sucrose will not undergo, do not undergo mutarotation is tendency to rotate within its. Whenever, you know, whether it is glucose, whether fructose, whatever sugar is dissolved, it forms a solution and it is allowed to rotate. Yes, invert sugar, sucrose is an invert sugar. So, but sucrose will not never undergo mutarotation. There is a reason behind that why sucrose is not. Okay, mutarotation is simply tendency to rotate within itself along the, again, functional carbon atom. Just remember this because that's what I have shown here. You can see that this open form can go to alpha form, can go to beta form, or the three forms are interconvertible with respect to one another. So this is a characteristic feature only of reducing sugar. Tendency to rotate within itself. If glucose wants to be in open chain form for some time, glucose will be in that form. Suppose if glucose wants to go in a stable conformation, it might change to beta form, it might change to alpha form. So again the angle of rotation will be different, but it will be stable. Okay, so this is what is mutarotation. Then another important property linked to this is OSA zone formation, a very important I should say property from examination point of view also. OSA zone formation. Now see, again I will just give you a very simplified version of OSA zone formation. What happens is, we simply take these reducing sugars, glucose, fructose, galactose, mannose, or you can say other disaccharides, whatever reducing sugars are there, right? What we do is, we make a solution of these reducing sugars and we add phenylhydrazine into it. Phenylhydrazine, hydrochloride with anhydrous sodium acetate, right? We keep everything in a test tube like this and we keep it in the water bath for 24 hours. Obviously, glucose, fructose, galactose, depending upon their functional groups, they are going to phenylhydrazine, C6H5NH2. Again, I am talking about chemistry. NHNH2, you know, water molecule will be lost. It is going to form a phenylhydrazone. Wherever functional group is free, it is going to form a glucosazone, fructosazone, galactosazone, maltosazone, so on. So this is what we are forming, what we are getting, because every molecule has a tendency to form crystals. This is basically acting just to dehydrate it, because we are keeping it in the water bath also for some 24 hours. So then what happens is, formation of crystals is there. And these crystals are then taken out and we put them on a slide and we observe them under the microscope. So obviously we have to properly tease the crystals so that we can visualize, and you will see that. I think you are going to perform this practical also in your first year. This is again a very interesting practical. So depending upon the shape of the crystals, we identify which sugar it is. So if I talk about glucosazone or glucose crystals, glucosazone or fructosazone, the shape of the crystals is needle-like. Or it's like bundle of hay. Okay. If we talk about the structure of galactose, galactose has thorny edge-like structure. Edge-like, or it's like crystals are thorny, like, you know, just say those thorns are there, just like that, pointed. Okay. Then if we talk about lactose or lactosazone, because lactose, abhi we have not discussed the structure of lactose, it is a disaccharide. But here we are focusing on the reducing sugars. So lactose also has, it also shows crystals, and the shape of the crystals of lactose would be powdery puff. Right. And then maltose, it is known as maltosazone, and maltose is sunflower petals. Sunflower like petal. Right. Right. So you can say that OSA zone formation is again a characteristic feature of reducing, very, be it glucosazone, be it fructosazone, galactosazone, lactosazone, maltosazone. Now, depending upon this finding, can anybody tell me what will be the shape of the crystals of sucrose? Now tell me. Yes, very good. No change, or it will not form crystals. Why it won't form crystals? Because sucrose will not form the zones. Reason क्या है? Sucrose is the only non-reduced sugar. Correct. So you can say that all reducing sugars will undergo mutarotation except sucrose. All reducing sugars will undergo osa-zone formation except sucrose. So these are very, very important properties of monosaccharides, or you can say reducing. This what we covered, anomers we covered, and epimers. Then after this, we come over to the oxidation and reduction properties of monosaccharides. Just say for example, if we talk about oxidation, up see oxidation, again I will just tell you about the chemistry part of this. You know, CHO, CHOH 4, 5 and 6. This is the structure of glucose. If glucose undergoes oxidation under mild oxidizing agent, mild means very light. You know, aldehyde group is more susceptible towards oxidation. जैसे अब all of you must have prepared for your, this UG NEET exam. उसमें you must have studied about ascent of series, conversion, descent of series. Alcohol to aldehyde to carboxylic acid, isn't it? And ulta is reduction. So we can say alcohol is oxidized to aldehyde, aldehyde is oxidized to oxalic acid. Here aldehyde is there, immediately it will be oxidized even in the presence of a mild oxidizing agent. We perform the reaction in strong oxidizing agents, both will be oxidized, right? अब question is when this reaction takes place in the body, what happens? क्योंकि एक चीज होती है in vivo, एक चीज होती है in vitro. So if we talk about mild oxidizing agents, simple, you can say aldonic acids will be produced. Gluconic acid, fructonic acid and so on. But in the presence of strong oxidizing agents, saccharic acid will be produced. Okay. Saccharic acid means both the groups will be oxidized. Glucaric acid is there, right? And now the question arises, we have to hide this. Means we have to oxidize this primary alcoholic group. That means we are not oxidizing this aldehyde. This can take place only in the body with the help of the enzymes. And that is what exactly happens here. Glucuronuric acid is produced. So, gluconic acid is produced when this group is oxidized. And this reaction takes place by a very, very important pathway in the body, which is known as uronic acid pathway, which is considered to be an alternative pathway of oxidation of glucose. And this pathway occurs in the body just to synthesize this glucouronic acid. ठीक है, अब most of the students might get confused between gluconic acid and glucouronic acid. I'll just write it here. Gluconic acid and glucouronic acid. वैसे ही just remember uronic acid means which is produced as a result of, that's why I have written the spelling also. Gluconic acid and glucouronic. See the spelling. Gluconic means aldonic acid which is under mild oxidizing agent. Glucouronic acid which is synthesized by a uronic acid pathway. Whenever we talk about any pathway, simply write the enzyme. So this reaction takes place in the presence of the enzyme. Okay? And glucouronic acid plays a very, very important role in our body. Very important role. It is considered to be one of the very important, I should say, detoxifying agent. Detoxify का मतलब क्या होता है? Detoxification. D means removal. Toxic means, toxic जो harmful होते हैं. So glucouronic acid helps in removal of the toxic substance, or it converts toxic into non-toxic, so that they can be excreted in. So slowly you will study in the metabolism that how this glucouronic acid is produced. And always remember there are so many similes in biochemistry. So don't get confused. I'll show you so many similes. Inulin, insulin is there, dextrin, dextrins is there. One live example is there in front of you, only gluconic acid and glucouronic acid. Yeah, so isn't it interesting? Right? So oxidation may just remember one thing, that glucouronic acid is produced with the help of enzymes and rest is in the presence of either mild oxidizing agents or strong oxidants. Clear? Then coming over to the second property, reduction. अब now see reduction के लिए always remember it is actually the enzyme, because enzymes I told you they are the ones which are catalyzing the reactions. So the enzyme is dehydrogenase. Okay. Now again I'll give you one very simple concept. If I talk about glucose, glucose is a aldohexose. So the enzyme responsible for reducing glucose will be aldol, either dehydrogenase or I can say aldol reductase. If I talk about fructose, again the enzyme will be reductase. If I talk about galactose, the enzyme will be reductase. I'll just write the short form AR. And similarly, if mannose is there, the enzyme is again aldol reductase and the product will be used. Okay. Now one very important point about this enzyme is this enzyme consists of zinc also sometimes. Zinc acts as a cofactor for reduction. Zinc is the major coenzyme, cofactor for dehydrogenase, but sometimes it acts with reductase. Okay. One more enzyme is there. Again, don't get confused with alcohol dehydrogenase. We will be, after chemistry of carbohydrates, we will be taking enzymes. Then I'll show you what is the basic. Yes, I am telling you, beta, the enzyme will be reductase. It's not ketal reductase, it will be keto reductase. It will be keto reductase, not ketal. Ketal is altogether a different product. Okay. So, alcohol dehydrogenase, we always write it as ADH. Okay. And aldol reductase is AR. Means glucose which has aldol. Aldol means OH actually. हम फ्रक्टोज को भी कह सकते हैं aldol reductase, क्योंकि if you look at the structure of fructose also have this type of structure, isn't it? OH तो है. Okay, so aldol is coming, aldol means it is coming from ol. Ol is coming from this OH. So even if we write it as aldol reductase, then also it is correct. So don't get confused. Okay, it is oxidation of this. So glucose and fructose are going to form sorbitol, right? Sorbitol. If we talk about galactose, galactose will form galactitol obviously, galactitol. But it is also known as dulcitol. In some books it is given as dulcitol. And mannose will form mannitol. Okay, so you can say here we are talking about reduction. So all the reducing sugars will have a tendency to undergo reduction in the presence of enzyme aldol reductase. Sorbitol, sorbitol, galactitol, mannitol. Now coming over to what is the significance of this. Because always remember one thing, that carbohydrates are the universal sources of energy. We are walking, we are talking, we are doing so many activities. So carbohydrates are the ones which are giving us the energy. Protein, because what all we eat in our diet, you know, carbohydrates we have, proteins we have, fats we have. So if we talk about the universal sources, carbohydrates are the ones. And you know, excess amount of carbohydrates are converted to fats also. And they are stored in the form of fats. Proteins are required for building up of the muscle mass, and fats are the instantaneous sources of energy, because even when fats break down, they form an intermediate which is more or less related to this only. Okay, so what is the significance of this? Up now see, we are having carbohydrates, we are having glucose, fructose, whatever we are having. Whenever there is increased concentration of glucose, obviously the concentration of this will increase. Fructose, this will increase. Galactose, this will increase. Mannose, this. Now, here the twist arises. If the concentration of glucose increases, remember this condition is known as hyperglycemia. Glycemia actually comes from the glycemic index basically. So hyper is increased concentration of glucose. Now see, whatever energy we are getting by the breakdown of glucose. Okay, so glucose is giving energy to the eye muscles also. Eye muscle is also contracting, blinking, so many functions. So what is happening is, if the concentration of glucose increases in the eye, obviously there will be increased concentration of sorbitol. Now just try to understand this. Okay, in case of hyperglycemia, what is happening is concentration of glucose is increasing, concentration of sorbitol also increase. Up the tragedy with this sorbitol is it cannot cross the eye membrane. Up when it cannot cross the eye membrane, its concentration will keep on increasing, keep on increasing. Sorbitol, it is an alcohol. It will increase the pressure inside, which is known as ocular. So what will happen is, it is like a balloon. You know, balloon mein when the level of water increases, it might burst all. Same is the case here. And that is what we say. During hyperglycemia or during diabetes mellitus, the worst effect is on the eye. The reason behind that is that there is increased concentration of sorbitol in the eye, which cannot cross the eye membrane, thereby increasing this ocular pressure, thereby leading to the formation of cataract also. It can be reversed. Again, I'll come to that if we give that patient inhibitors of aldol. But once the damage is done, it can be controlled, but it cannot be completely fixed. We can control it. Okay, so what happens is, if we give them these inhibitors, aldol reductase, what will happen? We'll try to minimize the concentration of sorbitol. But once it has increased, there may be, you know, the blood starts oozing out from the eyes also. So what we do is, we regress the blood vessels so that no more, you know, blood comes out. So the reason, the biochemical reason behind this is increased concentration of sorbitol, thereby increasing the ocular pressure. So most of the drugs which work on, they are aldol reductase inhibitors, which will try to minimize the level of sorbitol. Right. So this is one very important, you can say, reducing properties of, and the biochemical significance also of this. Coming over to galactose. Galactose also leads to formation of galactitol. Right. So abhi right now we will be discussing, when we will discuss the metabolism, I will tell you one very important metabolism, inborn error of metabolism of galactose, which is galactosemia. Semia is blood. Whenever the increased concentration of galactose is there in the blood. So what will happen again? There will be increase in the concentration. Yes, very good Nagesh, galactosemia. So what will happen is, there will be increased concentration of this galactitol. Again, there will be formation of. But again, we will come to that. The cataract formed by sorbitol and by galactitol will be different. There it is snowflakey cataract, here it is bilateral cataract. So we will be discussing when we will discuss the metabolism, right? So these are what? These are the reducing property. Then coming over to mannose, which is again a reducing sugar, it forms mannitol. Now, what is the significance of this mannitol? This mannitol decreases the intracranial pressure. So, in any injury, what will happen? This mannitol is given, so it will immediately decrease that pressure which is there to heavy flow of bleed. Right? So, this is what this is the reducing. So today what we have discussed in today's live session, let me just quickly summarize it. What we have discussed is, we have discussed the properties of monosaccharide, right? And what properties I have basically focused on? We have focused on anomers, differing at C1. We have discussed about epimers, differing at any other position except C1. Then we have discussed about reducing sugars. The reason why I am giving you the gist of the lecture, so that in case of any confusion, you can ask me now also. Reducing sugars we have discussed, which have free functional group. Okay. Then again non-reducing sugars also, and reducing sugars ka in next session we are going to discuss what is the biochemical significance of these reducing sugars. As just now I told you that this is a very, very important abnormal constituent of urine. Right. Then above all, we have studied about stereoisomers. And we have studied about optical isomers. Along with the properties oxidation and reduction. So I hope whatever we have discussed is clear to all of you. Timing beta, timing will be intimated to you. So in the next session, I will try to finish up with the entire chemistry of carbohydrate. So if you guys have any confusion, you can ask me. And if you have already come in a semi-drowsy condition, you can ask me in the next session also. Okay? Timing automatically will be, we will intimate you about the timings. So shall we wind up the class? Okay, my pleasure. Shall we wind up? Sanjay, Sagar. Yeah, my pleasure. So, yeah, thank you, Dr. Abhijit. So see you guys in the next class, and try to circulate about my class with your other friends also, so that maximum students can attend the class. Because it's like, you know, there is a saying in English, the more the merrier. So it will be an open discussion. So good night all of you. See you all in the next session. Bye-bye.